Does chiropractic manipulation affect the central nervous system? It's intriguing and appealing to imagine that there is something unique and special about what chiropractors do in contrast to standard medical care. For years it was postulated that the reason for so many unexplainable successes with chiropractic treatment was that chiropractic care not only, quote, normalized nerve function peripherally, but that the effects were more far-reaching, including directly affecting the brain and all of its complex functions, including modulation of the autonomic nervous system, mental function, immune function, and neural integration of movement, and therefore functional optimization. There's been a recent surge in a search for an objective demonstration of this effect through the use of sophisticated imaging and electrodiagnostic testing. Let's review what is known and what can and cannot be stated at this point regarding the evidence. We'll divide this discussion into two presentations, starting first with functional MRI basics and concerns. First, what is it that is being measured? Generally, the focus is on, are there changes in cerebral blood flow post-intervention? Are there specific areas of the brain that are activated with manipulation? And are there central nervous system reflex changes that indicate distal effects, for example, strength of muscle contraction? And a long-standing search continues to determine if there are any changes in autonomic nervous system activity due to chiropractic treatment, with measures including salivary amylase, pseudomotor activity, skin temperature, and heart rate variability. And of course, some of these are assumed indicators of local dysfunction and therefore are, quote, subluxation finders for some. Given the breadth and complexity of the topic area of autonomic effect, we will briefly indicate for the autonomic nervous system what the general consensus is at this point, but we'll then focus on the more recent attempts using functional imaging and electrodiagnostic testing to detect central nervous system activity after manipulation. A very recent systematic review and meta-analysis was published in Chiropractic and Manual Therapies entitled The Acute Effects of Joint Manipulative Techniques on Markers of Autonomic Nervous System Activity, a systematic review and meta-analysis of randomized, sham-controlled trials. The researchers conclude, quote, given the current state of the knowledge from randomized sham-controlled trials, spinal manipulation and mobilization with movement may have no acute effect on the studied markers of autonomic nervous system activity. Overall, the certainty of evidence was often assessed as very low or low, unquote. Now to the focus of this discussion, measuring central nervous system activity. Well, let's count the ways. The approaches include EEG, transcranial magnetic stimulation, PET scans, functional MRI, and electrodiagnostics including the H reflex and M and V waves. Regardless of approach, the questions that need answers include, what is the purpose of the measurement approach and what are its limitations? In other words, are these directly viewing physiological central nervous system changes such as vascular flow, or are they indirectly through a proxy suggestive of these changes? What is the outcome measure or outcome measures? Is it simply the results of the functional measurement approach, or are there other measures, including pain and function? How significant is the difference or change? And of course, this must be matched to a comparison group and/or a baseline measure of activity, and although the results may be statistically significant, would these reported effects be clinically significant? For example, how long does the effect last, and which chiropractic technique is being assessed? Generally, there are three main areas of investigation: the study of CNS stimulation to determine what effect spinal manipulative therapy has on specific CNS areas of function and process, function to determine the effect of SMT on muscle reaction and strength, and finally pain. What is the effect of SMT on pain threshold and reactions to pain stimulus? Given pain is often a co-measure in studies of central nervous system stimulation and/or function, we will include the discussion of pain together with these other measures. Let's start with central nervous system stimulation and a short review of the imaging approach, its pluses and minuses. It's important we review basic principles and what can be determined versus what can be inferred from areas of activation. Functional MRI has a higher spatial and temporal resolution compared to PET scanning and does not involve radioactive material. It detects increased blood flow to a brain region, and the amount of oxygenated blood is a proxy for increased activation of neurons. The relative concentrations of oxygenated to deoxygenated blood creates a signal known as BOLD, blood oxygen level dependent response. The higher the ratio, the higher the energy consumption. So how is brain activity measured? Well, we know that a pixel equals a two-dimensional measure of resolution on TVs and monitors. A voxel is a 3D measure. It's a volumetric pixel element measuring 3 cubic millimeters, representing a value on a grid in 3D space. There are approximately 40,000 voxels possible in the brain. Interpretation of a functional MRI requires a subtraction approach. Researchers compare scans to either a baseline comparison or between two scans of common tasks with one novel component. The common areas for each are canceled out as black, while the unique area is subtracted out as lighter. On a voxel-by-voxel basis, each is assigned a color dependent on the strength of the difference in activation between the control and experimental conditions, subtracting out the common areas of activation. In other words, the computer highlights the regions that become more active in one scenario versus its comparison scenario. The brighter the color, the greater the confidence of a difference. This is basically the p-value or confidence interval that the computer generates for a visual demonstration of the degree to which chance plays a role in the finding. Also, brain scans in studies are oftentimes the average or mean image of all the participants. There are some significant misconceptions with interpretation of the meaning of functional MRI results. Scientists can't read thoughts. They only can tell us what areas of the brain known to be associated with certain thoughts or feelings have demonstrated increased activity. These are neural correlates. Oversimplification that a specific area of the brain is solely responsible for a given neural function is naive. For example, Broca's area for language is not the only area involved, and it's part of a node or convergence center for language. And as one writer puts it, most neural real estate is zoned for mixed-use development. In this publication, The Relation Between Statistical Power and Inference on Functional MRI, researchers analyzed a real functional MRI dataset of nearly 500 subjects performing a social cognition task collected through the Human Connectome Project, which we will discuss later. To represent typical functional MRI studies, random samples of 15 subjects were drawn to assess within-subject effects, and random samples of 30 subjects were used for between-subject analysis. Results from these examples were compared to data from the full sample of 500 subjects to determine how strong the real effect is. Effect sizes negatively correlated with sample size. In other words, underpowered studies, those with a small number of participants, can result in grossly overestimated effects and inconsistent activations. The main concern expressed by many scientists is the marketing of functional MRI and a misrepresentation of what can be determined based on which areas light up on a functional MRI. In other words, assumptions of function related to behavior or disease. For example, the amygdala is synonymous with fearfulness. It reacts to photos of menacing facial expressions, but also becomes more active with happiness, anger, sexual arousal, unexpected and unfamiliar, exciting things, and registers the personal relevance of a stimulus at a given moment, for example, whether somebody is hungry or tired. And other common examples assumed to be related to behavior or mental function are that the dorsolateral prefrontal cortex is involved in the inhibition of one's spontaneous responses, so it's related to risk, reward, and attention, and dysfunctional in those with ADHD, autism, and other neurologic conditions. The ventral striatum is considered to be involved in reward and pleasure, but it's also reactive to novelty. And the insula, which is often simplified as activated as a reaction to aversion, has at least 10 anatomical subunits, each with its own specialized population of cells that play a role in various experiences. These include trust, insight, empathy, uncertainty, aversion, and disbelief. It also integrates visceral states with emotion, such as pain, hunger, thirst, and temperature. The hype of functional MRI's ability to see or visually display what is normal versus dysfunctional is being coined as neuromania, neurohubris, and neural hype, among others. Here are some examples of overestimation of value. In this article, This Is Your Brain on Politics, they suggested that functional MRI could determine or predict who would win the 2008 presidential election. Two candidates failed to, quote, light up or stimulate the brains of voters on the brain scan. They were John McCain and Barack Obama. And in addition to being increasingly used as a defense in criminal trials, there is a service marketed as No-Lie MRI. They market to employers that they can screen potential employees to see who will lie or not, and predict other unwanted behaviors based on correlated areas of the brain that light up with certain cues. Here's an entertaining and cautionary study on being overenthusiastic about functional MRI results. The title is Neural Correlates of Interspecies Perspective Taking in the Post-Mortem Atlantic Salmon. Quote, the salmon was shown a series of photographs depicting human individuals in social situations. The salmon was asked to determine what emotion the individual in the photo must have been experiencing. And, you guessed it, areas lit up in the functional MRI of this dead fish. Scientists warn about reverse inference. Functional MRI does not allow the conclusion that structure X causes function Y. It's only correlation. In other words, which area is active with specific activities, not which brain area causes a particular psychological reaction or behavior. Mental functions are rarely localized to one part of the brain. Even the link between blood flow and neural activity is not straightforward. There is a delay of 2 to 5 seconds between activation and increased oxygenated blood. Also, regions of inhibition may light up because the neurons that are inhibiting other neurons require energy. Voxels are still too big for spatial resolution for small clusters of neurons, and because the brain becomes more efficient with repetition, the blood oxygen levels may be lower on repeated performance or exposure. And in this Science Alert post, a bug in functional MRI software could invalidate 15 years of brain research. Quote, to test how good this software actually is, Eklund and his team gathered resting state functional MRI data from 499 healthy people sourced from databases around the world, split them into groups of 20, and measured them against each other to get 3 million random comparisons. They tested the 3 most popular functional MRI software packages. The software resulted in false positive rates of up to 70%. One of the bugs had been in the system for the past 15 years, which explains why so many papers could now be affected. The bug was corrected in May 2015, unquote. With all this discussion, it may appear there is no current feasible use for functional MRI. But the current use is for pre-surgical appraisal prior to neurosurgery to determine areas that may be resected without affecting function. It's the best available tool, even with its limitations. So it's probably prudent to think of functional MRI findings as reactions to stimuli in comparison to baseline levels of activation without that stimulus, rather than an indicator of mood, behavior, or pathology directly.